The exchange of the materials between the capillary and the tissues occurs across the endothelium by several different mechanisms. We'll be looking at these three mechanisms, beginning with diffusion, a passive transport mechanism where substances move from an area of high concentration to an area of low concentration. If a substance is hydrophobic, it can diffuse right across the plasma membrane. If it is hydrophilic, it will diffuse using transport proteins. Water will diffuse through protein pores called aquaporins. A second mechanism for the exchange of materials between the blood and the tissues is transcytosis, a form of active transport which requires ATP. Transcytosis involves pinocytosis on one side of the cell, where fluid with solutes are brought into the cell. They are then transported by way of transport vesicles to the other side of the cell and released by exocytosis. Transcytosis allows you to transport larger substances into, through, and out of the cell. The last mechanism is bulk flow. This is what we're going to investigate further. As the term bulk refers, we are moving large volumes, in this case, plasma and solutes, within the plasma. Bulk flow can move plasma with solutes from the capillary into the interstitial fluid surrounding the cells, and in the opposite direction from the interstitial fluid into the capillary. The fluids with their solutes can move through intercellular clefts, which are gaps between adjacent endothelial cells, or fenestrations. These are pores in the membranes of the endothelial cells. Bulk flow occurs by two mechanisms: filtration. In filtration, the movement of plasma is from the capillary into the interstitial fluid. Reabsorption is the opposite. This is the movement of interstitial fluid back into the capillary. So what was filtered is returned back to the blood. Bulk flow occurs along pressure gradients, in other words, from an area of high pressure to an area of low pressure, and we're going to measure those pressures in millimeters of mercury, just like you measure blood pressure. Let's look at these pressures. First, we'll begin with hydrostatic pressure. Hydro refers to fluid, so this is the pressure that is created by the fluid. The hydrostatic pressure within the capillary is actually the blood pressure in the capillary. We can use the abbreviation HP for hydrostatic pressure and the subscript C for capillary. This pressure pushes fluid out of the capillary, which is called filtration. Notice that the hydrostatic pressure in the capillary, or the capillary blood pressure, drops as the blood goes through the capillary. At the arterial end of the capillary, the beginning of the capillary, the blood pressure is 35 millimeters of mercury. But by the time the blood reaches the venial end of the capillary, it has dropped to 15 to 17 millimeters of mercury. The hydrostatic pressure in the interstitial fluid is 0 millimeters of mercury, so it is non-existent. We'll use the subscript IF for interstitial. Because this pressure is 0 millimeters of mercury, we're going to ignore it. So thus, the hydrostatic pressure or blood pressure in the capillary is responsible for the movement of plasma with the solids out of the capillary into the interstitial fluid, which is called filtration. A second type of pressure is osmotic pressure or colloid pressure. This is the pressure that is created by the solids. The solids will be proteins. Proteins in the capillary and proteins in the interstitial fluid. The fluid will move by osmosis from an area of high water to an area of low water. If you recall from AP1, solids determine how much water you have. There are more proteins in the capillary than there are in the interstitial fluid. Let's look at two types of osmotic pressure. The osmotic pressure in the capillary, produced by the plasma proteins. This osmotic pressure is 26 millimeters of mercury. Since plasma proteins are too large to move out of the blood vessel, this pressure will remain constant throughout the entire capillary. And it will draw water or fluid back into the capillary. We call this reabsorption. So this pressure will pull fluid into the capillary. The second type of osmotic pressure is the osmotic pressure of the interstitial fluid, and that is 1 millimeter of mercury. Since this is such a low pressure, we're going to ignore that pressure. What determines the movement of the fluid, whether you have filtration or reabsorption, is the difference between the osmotic pressure in the capillary and the hydrostatic pressure in the capillary. Let's look at this. Let's compare the hydrostatic pressure and the osmotic pressures in the capillary. Notice the hydrostatic pressure drops as the blood moves through the capillary from 35 millimeters of mercury to 17 or 15 millimeters of mercury. The osmotic pressure, on the other hand, has not changed. Recall that proteins are responsible for the osmotic pressure, and those plasma proteins are too large to leave the capillary. That pressure, in turn, will remain constant. At the arterial end, the hydrostatic pressure is greater than the osmotic pressure. And recall, hydrostatic pressure is responsible for filtration, osmotic pressure in the capillary for reabsorption. Since the hydrostatic pressure is larger, filtration will occur at the arterial end. At the venual end, notice that it is the osmotic pressure that is greater. Recall, osmotic pressure is responsible for reabsorption. The net filtration pressure is the pressure responsible for filtration. Recall, this is the hydrostatic pressure in the capillary. About 20 liters of plasma is filtered per day. We have 5 liters of blood, and of that, about 3 liters is plasma. So 20 liters is almost 7 times your plasma volume. You are filtering a lot of plasma every day. When that plasma with the solids moves into the interstitial fluid, that plasma has to go back into the capillary or your blood pressure would drop. So at the arterial end, and this is important, you need to know that filtration occurs at the arterial end of the capillary. You will filter this plasma with solids. But at the venial end, you are going to reabsorb the majority of it back. And this is going to help maintain your blood pressure. You'll notice that only 85 to 90% of what was filtered is reabsorbed. So what happens to the remaining 15 to 10%? It is taken up by a lymphatic capillary. Once it's in the lymphatic capillary, it becomes lymph. That lymph then returns to the blood at the subclavian veins to become plasma again. Edema is the abnormal accumulation of fluid in your tissues. Recall this tissue fluid is your interstitial fluid. And this leads to swelling. That is the edema. Edema can enlarge internal organs, which will interfere with their function. There are several causes for edema. The first being increased filtration due to elevated hydrostatic pressure or blood pressure in the capillary. This increases the amount of fluid that is filtered. The amount of blood in a blood vessel influences that vessel's blood pressure. An increase in blood volume will increase blood pressure. Blood flow into the capillary and thus blood pressure is regulated by vasoconstriction and dilation of the terminal arterioles and the precapillary sphincters. Another cause for edema is increased capillary permeability. This occurs during inflammation. Histamine, released by basophils, increases the permeability of your capillaries by making the intercellular clefts larger. This allows for more plasma with solids to be filtered, which leads to the swelling or the edema that we see in inflammation. In addition, you might have edema because you have decreased reabsorption. If your liver is not functioning properly, you will not be producing sufficient plasma proteins. Albumin is the main plasma protein. If you have a drop in plasma proteins, that means you have more water compared to the solids, therefore, more plasma will be filtered. A third cause for edema is obstruction of lymphatic drainage. If a patient has had cancer surgery, which involves the removal of lymphatics, the 10 to 15% that is taken up by the lymphatic capillaries will not occur and accumulate in the tissues. Increased edema can interfere with the exchange of waste and gases. In pulmonary edema, the respiratory gases cannot diffuse across the respiratory membrane, leading to low oxygen levels and elevated carbon dioxide levels. The lack of oxygen will lead to necrosis or tissue death. Edema can also lead to a drop in blood pressure. This is because decreased reabsorption will result in decreased plasma volume. Now it's time to check yourself. Answer the questions that follow, and then after you have answered those questions, you can go on to the next video. Take care.